pleasure and an honor to call on Professor Barry Schoub to actually talk to us, and everybody's here to listen to him, not to listen to me, nor to listen to the detailed introduction about his very distinguished past and about the work that he's been doing recently. Suffice to say that he is undoubtedly South Africa's most distinguished virologist. He's played a huge international role. And like many people, I suspect, on this call, the COVID-19 period has drawn on his expertise and his personal resources to make a contribution at a stage in his life when he might have thought that he was actually able to enjoy visiting his grandchildren rather than doing this kind of thing. So I'm delighted to hand over to him, and please put your questions into the Q&A box. We'll leave time for questions at the end. So Barry.
Yeah. Thank you very much, Professor Katz, for that very generous introduction. And good evening. I believe it's good afternoon. I'm not sure if it's good morning to some people that are viewing this. Good evening to all our colleagues. It's great being with you, and I hope that when I end this talk, that the mystery around this very enigmatic virus Omicron will be a little bit clearer. So without any further ado, I'm going to go straight onto my slides, because that's really how we do things. Okay, the Omicron variant itself is very close to our hearts in South Africa for a number of reasons. First of all, as you all know, it was first detected in South Africa. It was very intensively studied in South Africa. Like the beta variant before it, it also became a bit notorious. And Omicron got a bit of a bad name as the South African virus, with all the kind of consequences which followed from that. But let me go. It doesn't seem to be changing. I hope it hasn't infected my computer. David, shall I maybe come back in again?
Come back in again, if you don't mind.
Yeah. Maybe try this. There seem to be two screens. Let me see if this one's better. Doesn't seem to change. Oh gosh. Let me try the other one then.
You go out.
There are two screens here, and I don't know why. And
okay, you are allowed to share screen. Carry on. There you are.
Okay. Now it's working. Okay. Let me go through a brief timeline on Omicron as it appeared in South Africa. First of all, early in November, there were some indications, some signals from the routine diagnostic PCR test for COVID that there was something amiss. It was called the S gene target failure. I'll come to it a little bit later. Let me get my pointer up. I'll come to it a bit later, but this occurred early in November, early last month. And we did kind of have some inkling then that there was something afoot. It's actually quite interesting because this S gene target failure was actually picked up by a junior technologist in one of the private clinical laboratories, one of the lesser clinical laboratories in one of the kind of semi-rural areas. So it just kind of goes to show. But the importance of this is that this actually gave rise to a very easily diagnosable test for S genes. In other words, instead of doing the full genomic surveillance, you can use this S gene target failure to diagnose. At any rate, more and more cases then started appearing with this S gene target failure. And then on the 11th of November, the first genomic surveillance started, and it then became apparent that there was a new variant on the way. It first detected in Botswana, For those who don't know, it's a country in the northwest of South Africa. And then a day later in South Africa. This one probably did come from South Africa anyway, but first detected in Botswana. And then a little later, on the 24th of November, the first official report by South African scientists, and I must just point out that it was first announced by South African scientists. It was not announced by your Secretary of Health, Mr. Javid, and he apparently in the House of Commons, he said that anyway, there was a bit economic with the truth. It was first reported by South African scientists on the 24th of November, and 2 days later it was reported to the WHO, that who then designated Omicron, following the sequence of Greek letters. Omicron is the 15th letter in the Greek alphabet. By the way, it leaves another 9 for future variants, and then they'll have to look at another maybe then they'll go to the alphabet or some other designation. And this then became the 5th variant of concern after alpha, beta, gamma, and delta. The 5th variant of concern. 7th of December, of course, the much awaited laboratory neutralizing antibody tests were announced. And last week, in fact, the first vaccine efficacy studies were released by Discovery, which is the main medical aid system in South Africa. So what I really want to kind of point out is that these are actually fairly new studies. There's a new virus, and I'm just want to really use the immortal words of Donald Rumsfeld. As you remember, he was the Secretary of Defense in the Bush administration, George Bush administration. And he said, there are no knowns. These are things we know that we know. So we've learned quite a bit, even in this, 6 weeks or so since we've had this virus, an enormous amount of knowledge has accumulated about it. But nevertheless, there are known unknowns. That is to say, there are things that we know we don't know. So there are things that we can define, the questions of which we need to know the answers to. And I'll come to that during the course of this talk. And then lastly, there are also unknown unknowns. There are things we don't know. And the Omicron has really kind of caught all of us, particularly in the virological field. And I must say, I've been 4 decades in the medical virology field, and this has really been one of the most enigmatic of the enigmatic viruses. So there are a lot of things we don't know about the virus, and there are still many surprises still to come to us. So I've divided this talk into these chapters. I'll start off talking about the pandemic. We can call it epidemic in South Africa, because this is really where the Omicron started off. And I think it's quite important to kind of look at the context of the South African epidemic. Then I'll talk about Omicron itself. I'll talk about the virus, this extraordinary virus, this virus which really kind of startled and alarmed the virological world. And I'll give that in detail, why it was so. And then going on from that, its properties. How do these changes, these mutations, this enormous number of mutations, how does that translate into the 3 main characteristics that we're interested in? Its contagiousness or infectiousness or transmissibility, the immune evasion, the ability of the virus to escape from either natural immunity or vaccine-induced immunity, and how this then expresses itself clinically in terms of its virulence. And then I'll try and make sense of all of this with a roundup at the end. So let's first start with the epidemic in South Africa. I know there are a number of South African or ex-South African, so you can close your ears and eyes, but just to point out to those who are not South African, you might often hear the term Gauteng. Gauteng, and it's pronounced Gauteng, not Gauteng, is the province where Pretoria and Johannesburg are situated. And this is really where the epidemic, or a pandemic, in fact, started. Pretoria, the capital of the country, there's Johannesburg. This is actually the smallest province geographically, and it's the largest province population-wise. And there's Botswana in the northwest of the country. So it started here and is now spread throughout the country. So let's look at the epidemic in South Africa up to now. So our first wave occurred in the winter of last year, and that was due to the ancestral virus, the D614G. Then this was followed by the second wave due to the beta virus, which was notoriously called the South African virus. It was first detected in South Africa, well studied in South Africa, and notorious for its immune escape, but it didn't spread very far beyond South and East Africa. And then we had the third wave, which was up to now the most severe wave, and that's due to, like, worldwide, to the Delta variant. And then now we're in the trajectory of the fourth wave. And up to now, these waves have been fairly kind of cyclical and followed a fairly reproducible pattern, that the waves themselves would last about 75, 76 days, except for the third wave, which is a lot more prolonged. That was about 100 to 105 days. But also the interval between the waves is also fairly predictable. This was about 90 to 100 days between the waves, except for the onset of the fourth wave. And we'd actually predicted, and we tried to be fairly confident in our prediction, that the fourth wave would start probably in South Africa towards the end of this month and probably well into January. And as a result, we could give a lot of comfort to, for example, the youth camps to hold their camps, to the holiday makers and so on. And we were caught really unawares. I must mention that this was unpredictable, because this interval over here is far shorter than this. This is only about 30 to 40 days. Anyway, as you can see, the curve is now very, very steep, the upward trajectory. It reaches a level far higher than we've seen up to now. This actually is now, of course, much higher as well. And also what is very noteworthy is that if you look at the previous waves, the hospitalization numbers followed more or less the 7-day moving average of the cases. There's a bit of lag over here, but more or less followed the 7-day moving average of the cases. And similarly with with mortality, obviously lower. But when we come to the fourth wave, our current wave at the moment, there's quite a disjunction between the 7-day moving average of cases and hospitalization and deaths. I'll deal with this in more detail when I come to the clinical expression, but just to point it out now, that hospitalization and particularly deaths are far, far lower than one would expect from the number of cases, and unlike what was experienced in the previous waves. By all the parameters, there's a lot going on in South Africa with the fourth wave, despite being mainly clinically mild. If you look at parameters, for example, the percentage of tests which are positive, unfortunately, this red curve over here should go all the way up to 35%. It's the highest it's ever been. We've never had so many percentage positive of tests as we have now, indicating the extent of the epidemic. Look at another parameter, another sensitive parameter is wastewater surveillance. We've had quite a lot of experience with wastewater surveillance with the polio surveillance, so it was quite easy to actually plug it in to COVID surveillance. Tshwane is the current name for Pretoria, by the way. And it's really, the epidemic started in Pretoria, then moved to Johannesburg, and then to the rest of the country. And you can see again, there's a sharp increase in the volume of virus, or the viral load in wastewater, reflecting the amount of virus which is excreted into the sewage. So all these parameters showed us that there was a rapidly expanding, a very steep upward trajectory of cases. Now, let's look at the virus which is responsible for it, because when we did the genomic surveillance, the characterization of the sample, now, presently, virtually all viruses are Omicron. There's very little Delta, which is remnant, and a tiny amount of alpha virus present in the country. So it is Omicron-driven fourth wave. So this is the S gene target failure, which has now really become a diagnostic test. It's basically this particular PCR, it's a commercial PCR test, the Thermo Fisher TaqMan assay. It picks up it amplifies various genes in the virus. And what you notice that the S gene, which is the most critical gene, S stands for the spike protein. That's the outermost part of the virus that the virus uses to attach to receptor sites in the respiratory tract and then establish the infection. And what they found diagnostically, and initially, as I mentioned earlier, this junior technologist, but now all the clinical laboratories, was that the S gene failed to amplify in this particular test. So you'd get a PCR test positive, except for the S gene. And it's called the S gene target failure. And that increased very, very rapidly. So fortunately now, there's quite a slick diagnostic test, part of the PCR, to diagnose Omicron. Now, what we are very, very fortunate in South Africa is that we've got a very, very well-developed genomic surveillance network. The reason for this is that this is an outgrowth of the HIV surveillance, HIV molecular resources in the country. And it was then quite easy to kind of extend it into COVID genomic surveillance. So there are 7 virology laboratories around the country, and they are continually doing genomic surveillance. And we probably got one of the world's top, according to Dr. Tony Fauci, it's not myself who's saying that, Dr. Tony Fauci came out and said that we've got one of the world's most advanced genomic surveillance system for surveilling new variants. And they were very quick in the middle of November to pick up that there was a new variant. And this is just the chronology. The alpha variant, the beginning of the year, that was your variant in the United Kingdom. Then we had the beta variant, which was our variant in South Africa, the gamma variant in Brazil and Latin America, the Delta variant, which kicked off in probably the biggest epidemic in the world in India. You remember the earlier part of this year. And then Delta, of course, became the Superman of variants. It displaced all the other variants and became the dominant variant in the world. And now we have the Omicron variant. Unfortunately, somebody put a Botswana flag here, which may be a bit unfair. And that's where we are now, the 5th variant of concern. So just a few words about this Omicron, or scientifically the B.1.1.529 variant. What really startled, and I'm not saying it inadvisably, startled the virology world, we saw in this variant something we'd never seen before in a number of respects. First of all, the number of mutations is extraordinarily large. More than 50 mutations, probably now going closer to 60 mutations, have now been detected in this particular variant. But more importantly, in the S protein, that critical spike protein, there are now 37 detected and defined mutations. 15 of these are on the receptor binding domain, the very superficial part of that spike protein which attaches to the receptor site. And in fact, the receptor binding motif, that's the very, very tip of the receptor binding domain, there are 9 mutations. Now, if we look at these mutations, this is the schematic of the genome of the virus. It's a long RNA virus, which up to now we thought really in the early days before in 2020, we thought this would be a cinch. You know, we'd be able to design a vaccine, but I think where we really were caught completely unawares is that this is a highly mutable virus. Not as mutable as HIV or influenza, but certainly far more mutable than the other RNA viruses, the measles and polios and all the other ones. Now, many of these mutations are old friends of ours. The 417N, the 484A, the 501Y, these are mutations which govern both transmissibility, in other words, infectiousness, and also vaccine escape. And we know them from the alpha variant, from the beta variant, the gamma variant, the delta. So these are mutations which are well recognized, well understood, and well translatable to how the virus is going to behave. Then there are other mutations which are not found in the variants of concern, but we do know from studies what they do govern, what they do translate to. And then there are a number of variants, a number of mutations. There are 14 of these which have never been seen in any variant, never been seen in any SARS-CoV 2. So we've got a situation of a virus here which has got so many mutations, it's got novel mutations, and this is the big question mark. And also it's got a unique constellation of how these mutations are positioned, particularly in the S protein. So this is a comparison of the Omicron S protein, the spike protein, versus the Delta, the previous Superman of variants. And if you look at these couple of variants in Delta, and you compare it, and Delta was, as I say, the Superman variant, the most highly transmissible variant. And you look at Omicron, this is Swiss cheese. I mean, this is riddled with mutations. And you can see even the actual shape, it's distorted the shape of the S protein. So this then said, this imposed a huge question mark. How is this virus going to behave? Is it going to be super virulent? Is it going to be super transmissible? Is it going to escape immunity far more effectively? And lastly, before we leave off the virus, these green little dots here are the various lineages of the Delta virus, which was before the most worrying of the variants. You can see numerous lineages. But when you look at Omicron, it hasn't had time yet to develop lineages. But what's important is that this is coming off a completely independent evolutionary tree, completely unrelated to any of the other previous variants. You can see it on this different, here's another cladogram showing the Omicron as a separate, completely different branch of the original root of the evolutionary tree of the virus. So this raises many, many questions. How is this virus going to behave? And fortunately now we're starting to kind of understand it. But before I get to there, of course, the big question mark is where do these variants come from? How do they arise? As I said, it's a mutable virus. Now, this is an interesting study which was done by Farina Karim in the African Health Institute in Durban. And they looked at an individual, a more advanced HIV immunosuppressed individual. And they showed at various time points how various mutations arise because this individual, this particular individual, is partially immunosuppressed and is partially immune. And because this person is partially immune, they don't clear the virus. There's a remnant of a virus which is then selected for to be vaccine evasive and also to be transmissible. This was then followed by a number of studies in other immunosuppressed individuals who don't clear the virus. And again, they select for these variants. That's the one group of individuals who then propagate variants and then propagate Omicron, of course. The other group, and I should mention, of course, South Africa has the highest HIV population in the world, the largest HIV population in the world. Because not only HIV, it's other immunosuppressed individuals as well. But also the kind of bulk of unvaccinated individuals, particularly on the African continent, also are a source of generating these variants. And these variants are selected for to be an advantage, an evolutionary advantage, to be immune evasive and to be contagious. So let's look at these properties. There are three properties we want to kind of define and characterize: the transmissibility or contagiousness, the ability to evade the immune response, either following infection, what you call natural immunity, or vaccine-induced immunity, and also the clinical expression. How severe is the disease due to this virus? So we started answering these questions. First of all, the transmissibility. As I showed in a previous slide, this fourth wave, this is in a fairly early stage, in fact. You can see this is almost vertical climbing, much, much more transmissible than any of the other waves. In fact, the doubling time of the Omicron is about 1.5 days, as compared to just under 2 days of the first, second, and third waves. The doubling time, in other words, the number of new cases, the time it takes for them to double. If we look at the reproductive number is the R number is the number which defines the contagiousness. It's the number of susceptible individuals who are infected from an infectious individual source. And this reached over 3 in the case of Omicron, over 50% more than the Delta variant, and the highest it's ever been in South Africa. We've never had a reproductive number, an overall reproductive number, which has reached 3 in our previous waves. So this is by far the most contagious virus that we've experienced in South Africa to date. And it probably relates to, this is a schematic diagram of this is the spike protein, that's the receptor binding domain. This is the ACE2, the angiotensin converting enzyme 2 receptor site that the spike protein binds to. And various binding studies, what we call binding affinity studies, have been done. And you can see that the Omicron has got the highest binding affinity. This is the inverse of the binding affinity of any other of the variants. This over here, this PM S20, is an artificially constructed virus with all the mutations to make it the highest binding as an artificial. This is not a natural virus. And you see Omicron has got the highest binding of any of these variants, highest binding affinity to the ACE2 receptor site. In fact, they've shown that the Omicron can bind to mouse ACE2 dimer. So in other words, it's really got such a high affinity, and because of this high affinity of binding, you get a much higher viral load in the upper respiratory tract, and therefore a much higher contagiousness and a much greater facility for the virus to bind to ACE2 receptor sites. Now let's look at the immune evasion, the ability to evade natural and vaccine-induced immunity. And it didn't take long. It took a couple of weeks for these results to be announced. There are now, 5 of these? 1, 2, 3, 4, 5, no, 6, which have, well, actually, it's actually 4 because this comes from a commercial company. But these, in the laboratory, one can test for immune evasion either using a live virus assay. The problem with this is a very cumbersome test. It's got to be done under biocontainment conditions, or you can use a pseudovirus. In other words, an artificially constructed virus with a spike protein genetically engineered into it. The live virus probably is the one which most closely mimics the real thing. And Alex Segal was the first one to report, and he found, in fact, 47 times the reduction of neutralizing antibodies able to neutralize Omicron. 47 times. Another live virus assay here found 30 times. This one found 16 times. A lot more than the pseudovirus assay, but probably the live virus is the one that's just closest to the natural state. So we're getting well, I'll show you in the graphs over here, a very marked reduction in the ability of antibodies to neutralize the virus. So this is with Moderna virus. Spikevax is a Moderna virus. Homologous boosting. AstraZeneca homologous boosting. Heterologous boosting, you get a better response, but still quite a marked reduction with AstraZeneca and with Pfizer vaccine. Homologous Pfizer vaccine also right down, almost in fact to a wipeout. And yeah, we look at post-infection convalescent sera from alpha infection, a wipeout, almost a wipeout. The beta, similarly, almost a wipeout. Delta, almost a wipeout. Interesting enough, the superimmune, now what we mean by superimmune are individuals who've had the infection, the natural infection, as a convalescent, followed by vaccination, and any of the vaccines, by the way. Then, in fact, you get much less of a reduction of neutralizing activity. So actually, the best thing to have is to have a natural infection and follow it up with vaccination. This is an interesting study which is done not in the laboratory. This was done by observation in the real life by Juliet Pulliam and her colleagues down in Cape Town. And they show that, in fact, individuals that had a past infection, there's a 12% relative risk of getting reinfected with a beta, the beta infection during the beta wave. That was our second wave. A 9% relative risk in the third wave, which is the delta wave, but only a 25% relative risk. In other words, more than twice the beta, and near going on 3 times where Omicron reduction of prevention of infection with the Omicron. In other words, now in our 4th wave. So out in the natural environment, again, there is shown to be relatively little protection, or far less protection from prior infection when exposed to Omicron as compared to the other viruses, the other variants. Now, it is really the transmissibility and the immune evasion which then governs the fitness of the virus. What we mean by fitness of the virus is the ability of the virus to replicate in a particular environment. In other words, how that virus is going to take over, as it were, and displace the other variant. And we can see that it's a highly transmissible virus. We can see that it's a virus which does very effectively evade the immune system. And if you take these two into account, we really have a fit virus, a very, very fit virus. So it's not surprising, and this is in South Africa, that the Omicron has displaced and almost completely displaced Delta. This is Beta, this is Delta, and this is now Omicron. And the same thing is now happening all around the world. This is England, Scotland, Denmark, Belgium, where Omicron is now displacing Delta and will probably, we can't be totally certain, but probably displace Delta throughout the world. Well, I say probably because certainly we thought Beta would, but Beta didn't. And it's now been, there's only one continent where Omicron hasn't been found, and that's Antarctica. So over 90 countries now throughout the world have had Omicron to various amounts. And certainly in Europe, it appears to be completely displacing Delta. So let's now go on to the clinical expression. How is it? Now, I showed you this graph before to show the disjunction between hospitalization data, death data, and the 7-day moving average. And certainly early on in the Gauteng province where the epidemic started, we can show that there's quite a disjunction between patients compared to the Delta wave. You can see over here, patients in ICU, relatively few from Omicron, the Delta wave, quite a steep increase, and of course, patients on ventilators as well. Almost no patients on ventilators due to Omicron. And the same thing here on these graphs. I won't go through in detail in the interest of time, but the same thing, this disjunction between the number of cases and hospitalization and also death and more severe cases. Okay, let me. One of the early interesting observations was that in the previous waves, children were hardly at all affected. Very, very few were hospitalized. Very few died. Now there have been very few deaths, but certainly admissions to hospital, you can see in the very young, far more. This is the 4th wave, the Omicron, as compared to the 3rd wave, the Delta. Far more young children were admitted to hospital and also had more severe clinical cases. And that actually applies to all individuals below the age of 40. And of course, the reverse is the situation with Omicron in the older individuals, where the Delta is higher, as expected. And this may be a reflection of immunization. In South Africa, there's no immunization at all with children under the age of 12, and it was 11 downwards. And from 12 to 18, to the teenagers, the adolescents, they've only had 1 dose of vaccine, of Pfizer, as yet. Whereas obviously above that, there's a reasonably high coverage, up to about 40% in the adult population. And this probably just reflects vaccination coverage. Just to mention where we are with vaccine coverage in South Africa, 39% of adults above the age of 18 have been fully vaccinated. 44% either fully vaccinated or partly vaccinated with one dose of Pfizer. The total population, 26 and 29%. In the teenage group, it's very small. It's only 14% so far been vaccinated, and relatively few individuals, and these are immunosuppressed individuals, have had a third dose, about 6,500. But what's very interesting in South Africa is the seroprevalence studies. Early on, our seroprevalence certainly was much higher, even at 24% and 40% in either random samples, such as blood transfusion or shopping center samples, or household surveys. It got up to about 64% with pregnant women, many of whom are HIV positive. But then more recently, and I say recently, this is to September, these figures are probably even higher now in December, but you can see these are various blood drops going up to September. There's a progressive increase in all ages, in fact, up to about 70% of the population. And in some samples now, up to 80% of the population have got antibodies to the spike protein. So presumably the majority of those are protective, at least partially protective. So this is a combination of the vaccine, which coverage is not that great, as well as natural infection. So we believe that natural infection in South Africa has been largely responsible for these very high levels of seroprevalence of antibodies. And that may well be a factor why the disease manifestation is relatively mild. So let me try and make sense of all of this. Let me round this all up and try and put this all into a kind of a logical, rational perspective of where we are now. First of all, let's look at the virus itself. It's a highly transmissible virus, the most transmissible virus, at least 50% more transmissible than Delta, and going to the ancestral viruses, probably 150% to 200% more transmissible. So it just shows you how this virus, with all these mutations, has moved along to become the most transmissible of all the variants up to now. Immune evasion, very, very significant evasion of immunity, both to the vaccine as well as to the natural infection, the post-infection convalescent natural infection. And this may well be why we're seeing, because of our high seroprevalence, clinical expression, which is relatively mild. But if you look at clinical expression, let's look at the pandemic itself. This is a well-known, this comes from the Johns Hopkins website. I'm sure many of you have seen it. Really, particularly in Europe, where many of you are, this really is where the bulk of the pandemic is. The South, even though we are well into our fourth wave, very, very much less as compared to what's seen in Europe. Both in terms, well, I'll go now, comparing the United Kingdom versus South Africa. The populations are more or less the same, a bit higher, of course, in the United Kingdom. But if we look at the 7-day moving average of daily cases, much higher, nearly 3 times as high in the United Kingdom as in South Africa. The daily cases adjusted to per million, again, 2.5 times in the United Kingdom compared to South Africa. And the same, I think, would apply to other European countries as well. The daily deaths as well, much higher in the United Kingdom than in South Africa, 29 versus 115, or daily 1.7 is against 0.5. And again, the percentage of infections which do result in mortality significantly higher in the United Kingdom than in South Africa. The vaccine coverage much higher in the United Kingdom, even though the cases and the severity of cases is much higher in the United Kingdom as compared to South Africa. A very important thing, I'll come to it later, is the median age. This is a very interesting graph because it shows you Denmark, United Kingdom, Norway, and South Africa in comparison. Now, Denmark, in fact, is the country which got the highest per capita infection in the world currently. It is also the country which has got one of the highest vaccine coverage in the world. And United Kingdom is like in between, and South Africa, which has got a relatively low vaccine coverage, but also much lower cases per million population. So let's try and analyze this. The one thing, of course, if you look at the tests that are performed, Denmark has got the highest tests per 1,000 population, 34.79, as against 1.19 in South Africa. So is the kind of relatively low, comparing the lower south compared to the north, is it maybe an artifact of the testing performance? That may be one possibility. Then if we come to the hospital admissions, this is the national hospital admissions, what's called DATCOV, data for covariance, for COVID. You see that we are increasing, but certainly nothing like in the previous waves, 7,339 as compared to 14,702. And also if we look at the severity, only 14% of these hospital admissions either on high care or intensive care. Now, one of the kind of problems in this, and I've pointed it out to them, are the incidentals. Because what happens is that people that are admitted into South Africa, South African hospitals, have to routinely get tested. And for example, pregnant women get routinely tested, and they would be part of these figures. There is on the surveillance form, there is a place which says reason for admission. So we try to get rid of the incidentals. Obviously, if someone comes in with a motor vehicle accident, they won't be part of these admissions. But there's a lot of gray areas, as you can imagine. And so these figures are contaminated to some extent by the incidentals. Then if we look at the deaths, and this has often come up, these are the excess deaths which correspond to the waves, whereas the notified COVID deaths are much lower. Estimated, the true deaths are estimated to be about triple what are officially regarded as COVID deaths. Now, why I'm pointing all of these out is that it could well be that what we are saying, the relative mildness of South Africa as compared to the global north might be artifactual. That may be a contributing factor. But it may also be real. And if it is real, what could contribute? Well, one of the things, obviously, an obvious difference is the median age. South Africa has got a considerably younger population than European countries, like the United Kingdom. 27.6 versus 40.5. And we know that COVID infection, this is all the way since 2020, is much more serious in the older population, much milder in the young population. So that may be a factor to a genuine mildness of the infection. Also, of course, the extent of antibodies may also attenuate the clinical expression of the mildness. So let's get back to Donald Rumsfeld. I've said a lot about the known knowns, and we've learned a lot about the known knowns about Omicron. There are known unknowns. We've defined, is it in fact truly mild? Is Omicron really a milder virus? Do all these mutations, in fact, not make the virus more serious, but in fact may attenuate the virus? Now, I want to not give the misunderstanding that viruses do mutate to become less virulent. That is a nice legend, but there's no virological evidence for that. These viruses mutate randomly, but there are the known unknowns, and that the big question we still need to answer, is it a tamer virus that just randomly mutated to become a tamer virus? And I think I wanted to use this quote from Arthur Conan Doyle. a capital mistake to theorize before one has data. And unfortunately, we don't have the data yet to say, is it a milder virus? Insensibly, one begins to twist facts to suit theories instead of theories to suit facts. So I'll end off with this, but there are also the unknown unknowns. The unknown, what's really going to come in the future? Omicron surprised us. Omicron was unpredictable. What's lurking behind the other variants in the future? Those are the unknown unknowns. There are things we don't know, we don't know. And I'll end off there. Thank you very much, and leave some time for questions and discussion. Hope I haven't overstepped the mark.
Thank you very much. You certainly haven't overstepped the mark, and you generated a huge amount of food for thought. I've been jotting down questions as you've been talking, and I wonder. One of the questions that is coming up is the question of underreporting, variations of reporting, variations in reporting of testing, as well as reporting of morbidity and of hospital admissions. How much of that is contributing, and are we seeing a scale of different countries in that at that level?
I think very, very definitely. I think underreporting must play a very significant role. I think, as I showed in the previous slide, testing in South Africa, we do now almost no test and trace. We tried to in the early days in 2020, it just was very, very difficult. And in fact, it has all but been abandoned now. So what gets notified is from the laboratories, from the clinicians, that what is known and usually are the clinically symptomatic, usually mild, but symptomatic cases. We really don't know what the iceberg of asymptomatic is. And I think that certainly must be a major contributing factor to the data in South Africa, for example. With regard to the deaths, the Medical Research Council have come out quite strongly that we quite markedly, quite significantly, quite grossly underreporting our mortality. We do know from the excess death data that the deaths are probably about triple what gets reported. So yes, I think underreporting is a very significant. And I think probably in Europe, European countries, there's a much greater test and trace, much greater random testing. And I think that the asymptomatic tests are much more thoroughly, the data is much more thoroughly obtained from Europe, which probably does account for the differences to a large extent.
The other question that has come up is, in your graph of Delta versus shifting to Omicron.
Yeah.
Somewhere along the line, are there dual viral infections? Are there ones with Delta plus Omicron? Or how's that happening?
I think those are the very odd cases which do get reported, and very, very few. As far as I know, we've never seen a dual infection in South Africa. It certainly has been reported in other countries, and it usually ends up being reported in the literature. We've never seen that in South Africa, as far as I know.
Now, when you say these are mutations, one of the questions is whether this particular one is a recombination, with a cluster of mutations in it rather than individual mutations.
Yeah, that's an interesting question. That has come up. Is it recombinant? Is it a recombinant virus? The only thing is this: there are two things against it. The one is that firstly, there are novel mutations. There are some mutations which have never been seen at all. There are some mutations which have been seen, but not seen in the other variants. In other words, there are mutations which have been seen, and mutations which should not lend itself to recombinants. For example, the mu variants, which are the lambda variants, the C12 variant. All of those are variants which don't occur simultaneously with Omicron, as far as we know. If Omicron did originate in Africa. And also I think the other thing against it is that it comes off a completely independent evolutionary tree. So I think all those factors mitigate it being a recombinant. I think these are spontaneous mutations. Why there are so many probably is a host factor. The partially immunosuppressed individuals who are harboring, and we studied that one which harbored for a couple of months. There may well have been individuals who are harboring for maybe even a year and a half, and that gives it ample time for viruses to accumulate all these mutations.
Right. Is the testing for Omicron effective? Are all the tests that the companies that produce, are they all detecting Omicron equally successfully?
Yes, well, all those which land up in PCR. Obviously, the point of care test, the antigen detection, you won't pick it up at all. But all those which land up at PCR, because now they're all going on to this this particular TACMAN test, which does pick up the S gene target failure, and the S gene target failure test has now become sort of a kind of routine as part of the PCR.
So in other words, the international testing is actually quite robust, from what you're suggesting.
Yes, I think that this S gene target. I mentioned the one commercial test. I think there are a few now which do pick up the S gene target failure, and that has greatly facilitated the diagnosis. Yeah,
there are questions that have arisen about, for example, if you have a little, not very adverse effect on children, and the vaccination doesn't prevent transmission, does one then need to vaccinate children? I mean, that's one way of putting the question. The other way of putting the question is to say, well, if the vaccine, you should really allow yourself to be infected with Omicron after you've had 2 doses of vaccine, because that will give you the best immunization.
Yeah, these questions do come up quite regularly. First of all, I think with regard to children, children, with classic COVID, they were thought to be very, very poor transmitters. I think Omicron being such a highly transmissible virus, that's changed quite a bit. I think children still significantly less than adults, that is true, but still significant transmitters because of the high transmissibility of Omicron. Children, their ACE2 receptors are immature, and that was thought to be one of the reasons why they don't transmit. But now, because you've got such a high binding affinity of Omicron, it doesn't need so many receptors to establish an infection. So I think children are infected quite really. They do clinically have a much milder disease. They do recover much quicker. Even those that are admitted to hospital, they do recover much more quickly than the adults. And very few of them, very, very few land up in ICU. But with regard to their transmission, they do still transmit. That is the one thing. And the other thing, children's habits are not exactly the most hygienic, and they don't wear masks very readily, and they do circulate very readily. So even if they are they're less good transmitters, put it that way, they are still at risk. And vaccination, of course, doesn't completely abolish transmission. It's not 100% effective, but it's pretty effective in reducing transmission. So I think there is a case to be made for vaccinating even very young children for that reason. With regard to deliberately or purposely infecting oneself with Omicron, it's a risky venture. It's a risky venture for 2 reasons. The one reason that, there are people that are seriously ill, and there are deaths. In South Africa, we're averaging about 50 to 60 deaths per day. Admittedly, the majority of those are unvaccinated, but there are some vaccinated which have died. So it's a risky venture to try and deliberately infect yourself. That's the one thing. The second thing is that you then become a carrier, and you can set up a chain of transmission and transmit to somebody that is vulnerable. So I think that's not such a great idea as well. And the third thing, which we don't know, because Omicron is still new, we don't know the risk of long COVID. In other words, the long disease, the prolonged disease, the side effects of Omicron. That still hasn't really been totally delineated. So you take a risk of getting long COVID. Now, admittedly, the long COVID with respiratory difficulties is extremely unlikely because the acute infection doesn't give you that. But maybe there may well be unknowns that we don't know in terms of a more prolonged clinical expression other than pulmonary expression. So I think for those three reasons, I think this wouldn't be a wise move.
Right. Along similar kind of lines, there's the question of asking it the other way. Isn't the reason why Omicron might be perceived as milder, at least in the UK, because you've had bouts of previous viruses, and bouts of vaccine?
Correct. No, I would think so. Yes. In fact, this in South Africa, this may well be why we got such a high level of seroprevalence, because the previous COVID viruses have circulated in a younger population, in a population which doesn't always have access to healthcare, and also a lot of overcrowding, particularly in the poorer areas, and that has spread the virus very rapidly. Most of them asymptomatically, many of them mildly, who didn't even bother to go for healthcare. And that is probably why we, to some extent, are fortunate because of a high level of natural infection.
Right. There are a couple of more questions, if you can bear with me. There are a couple of more questions on basic things like that, and then there are questions on therapeutics as well. So if I just, one of the basic ones is that the climate influence that there is. The mythology in the UK is that, it's going to peak in winter, and we're repeating last Christmas, whereas you appear to have shown quite clearly that the climate at the time of the infection doesn't affect the way
I don't think it does. We had our second wave in summer, in midsummer. It was December, which in Southern Hemisphere was summer, December, January. And the second wave was the second most serious. Our third wave was the most serious, and admittedly that was in winter. But it may indirectly, in that people are more likely to be indoors with poor ventilation. They're less likely to have open windows, open doors. When they're in taxis, for example, you won't get them to open the windows of a taxi in midwinter. You can forget about that. And they're less likely to have outdoor functions. And I think that may indirectly affect and make it more likely that you're going to get a more severe in winter. But certainly it's going to do with the virus. It's to do with people's behavior.
You've mentioned HIV immunosuppression being important in South Africa.
Yeah.
Of course, is that going to be different to the immunosuppression we see that's iatrogenic, or is caused by immunological disease?
No. In fact, that one slide I showed, it had those 4 studies where they showed the first one, the original one, was an HIV immunosuppressed individual, but the other 3 weren't. They were in people on cancer therapy.
Right. So at the immunosuppressed level, it's not going to be affected. In the pathogenesis, it might have been a contributing factor in
Correct.
In South Africa. And one of the basic questions that's actually come up with now is the question of the pulmonary inflammation. Maybe the pulmonary inflammation will take slightly longer. What are the risks of secondary infection superimposed on the pulmonary infection, even if you've got a mild form of lung condition, as in the 1918 flu?
Yeah, I think our view of the, and I think it kind of gels with the vaccination. The vaccination gives you systemic immunity. It's given in the arm, gives you systemic immunity. The only mucosal immunity you get vaccine, from the injection, is the IgG, which leaches out into the secretions. You don't get a significant IgA response. It's the passively leached out IgG antibodies. And this is one of the kind of advances, or our kind of next generation vaccine, what you're looking at. Vaccines given systemic, onto the surface directly, either by spray or orally, which will simulate mucosal immunity. And the kind of feeling in the pathogenesis is that the mild infections and the asymptomatic infections are localized to the upper respiratory tract. And the symptoms are due to all the chemical mediators, which you can speak to much more better than I can speak to them, the interferons and all those chemical mediators, which are responsible for the mild disease clinical manifestations. Once it gets into the lower respiratory tract, and that's why the vaccines are very effective. Any vaccines, all the vaccines are very effective to preventing the severe disease, because that's the systemic manifestation where you get very good systemic immunity, irrespective of whatever vaccine you're talking about. And that's why the vaccines don't give such good protection against mild disease, because you're relying on that passive IgG response. Yeah.
Thank you. I'd forgotten one, which is actually also frequently raised, is the question of, you showed the polio data. So this is from the water was the reason why you had this good information, good methodology. The survival on surfaces, it's always been, do these different viruses survive differently on surfaces in different kinds of ways?
The survival on surfaces was done very early on. So that's that, it was done with the original D614G, the original ancestral strain. Actually, if I say ancestral strain, to be more accurate, the ancestral strain is the Wuhan strain. The 614G was actually a mutant of that. But still, we talk about it as the original strains. They're not, 614G wasn't a very of concern. And the studies were done on those early viruses. Since then, because we know that epidemiological surfaces don't play any significant role at all, and I think nobody's even bothered to do surface survival. It was of interest in the early days. This virus is a respiratory droplet or respiratory aerosol, and there's a difference between the two: aerosol and droplet transmissible virus, and it goes from a person that's infected to a susceptible individuals by the generated droplets or aerosols. That is how it's transmitted. Surfaces play probably almost no role at all.
It's quite interesting that in the context that the one question that has come up must be from one of our virologists is there a difference in the structure of the spike proteins? Is anything known or been done about the actual structure of the spike protein with Omicron as yet, or is it too early to predictions?
I don't follow the question about the structure. You mean modify or using it as a vaccine or modifying?
No, the actual structure. Does the spike protein look different?
Yes. In that one slide, I know. I had the two together, and they did look morphologically different.
Yes.
Because the mutation do distort it, and that structure may well, and probably does, affect the binding affinity, because it binds much more effectively, not only to the human ACE2, but also to the mouse one. That's the only human variant which has been shown it's the only human variant to bind to a non-human ACE2 receptor.
Right, okay. Which leads one on to the first therapeutic question was, maybe ACE2 inhibitors should be reconsidered as a potential.
Yeah, as I think I mentioned earlier, I'm not a clinician. I'm not involved with therapy with management of patients. But yes, ACE2 inhibitors, initially it worked the other way around. And I must confess to being, how when one hears these things on the media, and that one Conan Doyle quote I said, you have to base it on facts. But I must admit to being taken by the media right in the beginning, because I was actually on an ACE2 inhibitor, and I stopped it because I was worried, maybe more susceptible rather than the other way around. So I stopped it for a while to my, shall I say, negative, because I didn't, the replacement didn't work well with me. I've now gone back to the ACE2 inhibitor. There have been a number of studies now where they've shown on individuals that are on chronic ACE2 receptor therapy, it doesn't affect them either way. It doesn't increase and it doesn't decrease their susceptibility. It seems to be completely unrelated on a number of very large studies which have been done.
Now, there are questions about Ronapreve and about, do you have any or of cocktails of monoclonal antibodies? Is there any impact?
Yeah, monoclonal antibodies are not used very, very little in this country. From expense is the one thing, and they're very expensive in South Africa. I think the kind of therapeutic, certainly from the publications, I don't treat patients myself, so I can only go on publications. And the kind of literature doesn't seem to be all that enthusiastic with monoclonal antibody therapy. Certainly been a lot more kind of enthusiastic for the molnupiravir, the Merck drug, and the Pfizer drug, whose name I always forget, the Pfizer drug, and the Merck drug. There seems to be a lot of promise certainly for treating early, because these are protease inhibitors and then protease inhibitors in terms of the molnupiravir. I can't remember what the site of the Pfizer one is, but certainly in the replication of the virus, they are direct antiviral agents. And the earlier they are used, the more effective they're going to be. There's very little point in giving them once you get the acute respiratory distress syndrome. There you've got to go to immunosuppressives like steroids, dextromethasone, and so on. But in the early stages, these drugs are showing a lot of promise in the literature.
Yeah, and you think any other therapeutic ideas, any other future therapeutic ideas that have occurred to you? I mean, that's the kind of question that I hate answering. So.
Well, our clinicians, and I'm rather a little bit embarrassed to say, they often kind of treat their patients with cocktails of vitamins and zinc, and I don't think there's really any kind of literature to support that. But they do it anyway, and there's nothing to lose, because I don't think any of these things are harmful, but I don't think they're effective. So certainly, in the early phase when you get viral replication, I think the kind of treatment has got to be an antiviral. There was a lot of, in the early days, I thought remdesivir would be the answer, and then they tried the anti-HIVs, and I think the one which is now showing promise are the molnupiravir and the Pfizer drug. And that may be effective. Then obviously in the later stages, the immune-mediated disease, it's the steroids which are
right.
main force.
I think that I'm conscious of the time in South Africa, and I'm therefore conscious of the fact that we've actually taken up a huge amount of your time. You've put in work to preparing this, as well as under huge strain of pressure of work in South Africa. And you've taken in a huge amount of time answering numerous different kinds of questions that we've put to you.
Thank you.
I want to thank you very much for this, and for the insights you've given, and for the insights you've given that are important not only in the South African context, but you've placed them in the worldwide context as well. I want to acknowledge on the program that, of course, on the webinar, that want to appreciation to Jonathan Brody, who facilitated the connection between us, and your wife, who facilitated the connection between us, so that we can get this going in absolutely record time, and and under the pressure and strains that everybody in the medical community and medical profession all over the world is feeling. So thank you very much, and we wish you all the best for a healthy 2022.
Thank you. And you too. Thank you very much for the invitation. I greatly appreciate it. Thank you.